Until the 1950s, most people thought genes resembled beads in a string. That is, genes were indivisible, and crossing over could only occur between the genes. After Watson and Crick published their DNA model, Seymour Benzer realized that if a gene were a sequence of bases, it could be divisible at many different locations, and crossing over could occur within a gene. In Bead's theory, crossing over occurs between gene E and F, whereas actually crossing over occurs within gene E too. Benzer demonstrated the gene's divisibility by crossing different virus mutants. Each mutant came from a different viral culture, but all had a defective R2 gene. After both mutants inject their DNA into a bacteria cell, some DNA from mutant number 104 crosses over with DNA from mutant 51, and the bacterium replicates the viral DNA. In the gene as a bead theory, all of the recombinant DNA would get a defective R2 gene. But Benzer found recombinant DNA that did not have a mutated R2 gene. This happened because DNA crossed over in the middle of the R2 gene. In this example, crossing over occurred between the two adenines in the R2 gene. The recombinant that didn't receive the mutations from its parents ended up with a wild-type R2 gene. Benzer made a map of the location of these mutations in the R2 gene by counting the number of wild-type offspring produced in many different crosses. The same method Alfred Sturtevant used to map the location of different genes in fruit flies. With this map of mutations in the R2 gene, Benzer tried to discover the minimum number of altered nucleotides that is required to make one mutation. Benzer's map, combined with other work, showed that only one nucleotide change is needed to make a mutation. These single changes are called point mutations. For example, mutant 104 has a point mutation where adenine replaces cytosine. Point mutations cause only one amino acid change in the protein made from the gene. Other base pair changes cause frameshift mutations. Insertions or deletions of one or more bases alter the reading frame in the DNA sequence and cause changes in many amino acids.